Hey class, welcome to week 2, or the chemical level of organization. We're going to start with some vocabulary as we do every week. So chemistry is the science of the structure and interactions of matter. And we are going to talk a lot about how different molecules interact and sustain life. Matter is anything that occupies space and has mass. And mass is the amount of matter in any object. And mass stays the same regardless of gravity. Whereas weight is the force of gravity acting on matter. So your weight could be different on Earth than it is in space because gravity is different. So the pull on your body is different. However, your mass will stay the same. Atoms are the smallest units of matter, and they are what make up our molecules and then our cells and all of the things that make us who we are. So they combine and interact in different ways, but they are the smallest stable units of matter. Each atom has 3 major particles. These are called subatomic particles. So these are particles that make up a single atom. So they're made of protons, neutrons, and electrons. Here you can see a hydrogen atom. You see the proton in the center. The proton is positively charged. Electrons are negatively charged, and they sort of rotate or spin around the proton in what's called an electron shell. And then neutrons are neutrally charged, so they do not have a charge. They're not positive or negative. So like I said, protons are positively charged. They are one mass unit in the nucleus. So if you have 1 proton, then you are 1 mass unit. Neutrons are neutrally charged, and they are also 1 mass unit. And most particles have, or most atoms have the same number of protons and neutrons. And then electrons are negatively charged, and they are low in mass. So they're very, very small. And they travel around the nucleus at really high speeds, spinning in what's called an electron cloud. And that cloud is made of specific electron shells. So the shells are at individual distances away from the proton and neutrons. And the outermost shell is called the valence shell. And the number of electrons in that valence shell determines how likely an atom is to form a bond with another atom, and therefore forming molecules. So like I said, the chemical properties of an atom are based on the number of electrons in the valence shell. So if the valence shell is full, then an atom is not going to be very reactive. Whereas if the valence shell is not full, then the atom is going to be quite reactive and want to go and bond with other atoms. Typically, atoms are electrically neutral, meaning that the number of electrons and the number of protons balance each other. However, if electrons are donated or accepted from other atoms, that's when we start to have charges to our atoms. So the first shell, the first electron shell, contains 2 electrons. And that's going to fill first. And then the outermost shell is called the valence shell. And like we said before, that determines the bonding. So the first shell holds 2 electrons. The second shell can hold up to 8 electrons. And the third shell can hold up to 18 electrons. Now the OpenStax book says that all shells after the first shell hold 8 electrons. That is not correct. So if you're using OpenStax, then do know that each shell holds more and more electrons. So if we look at hydrogen and helium, hydrogen, like we just saw, has one proton and one electron. So it's got one electron in its first shell, which would actually also be its valence shell. So that's going to make that pretty reactive, right? Because it's not a full shell. Whereas helium has two protons, and you can see it has two neutrons, and it has two electrons. So this is a full valence shell, and helium is going to be less reactive than hydrogen because it has a full valence shell. Lithium here has three protons and four neutrons, and it has three electrons. So it's got a full first shell and then one electron in its valence shell. So that's going to be highly reactive. Whereas neon has 10 protons and 10 electrons. So it has a full first shell of 2 electrons and a full valence shell of 8 electrons, the second shell. So the neon is not going to be very reactive. So hopefully you're seeing a pattern here. If the valence shell is full, it's going to be a less reactive atom. If the valence shell is not full, the atom is going to be more likely to react. A chemical element is a pure substance that's distinguished from all other matter by the fact that it cannot be created or broken down by ordinary chemical means. So these must come from the environment. Elements are designated by chemical symbols, like hydrogen has an H, carbon is a C, oxygen is an O, nitrogen is an N, as you can see here. And these comprise about 96% of the body's mass. If we break down the body, oxygen comprises 65% of the body, carbon is 18.5%, hydrogen is 9.5%, nitrogen is 3.2%, calcium is 1.5%, and then we get smaller and smaller from there. A couple more vocabulary words here. Inert means an element that does not readily react, does not readily participate in chemical reactions. So those are the ones that have the full valence shells. So they do not need extra electrons to make them stable. They are stable on their own. Reactive are elements that do not have a full valence shell, and so they are looking to achieve stability by gaining electrons or losing electrons or even sharing electrons with other atoms. And this is where chemical bonding takes place and molecules are formed. Molecules are when two or more atoms are joined together by strong bonds. And compounds are similar, but they are when two or more atoms of different elements are joined together by strong or weak bonds. So all compounds are molecules, but not all molecules are compounds. So if we see here, hydrogen, H2, is two hydrogen atoms bonded together. So that makes a molecule because it's two atoms bonded together. But it's not a compound because it's two atoms of the same, two of the same atoms. Whereas water is a molecule because it's two hydrogen and an oxygen, right? So it's three atoms bound together, but it's also a compound because it's two different types of atoms, hydrogen and oxygen. We have three different types of chemical bonds that we're going to talk about. We have ionic bonds, covalent bonds, and hydrogen bonds. Ionic bonds are bonds between atoms or molecules carrying an electric charge. So here, another couple of vocab words. A cation, which is here, is a positively charged ion. and an anion is a negatively charged ion. So if we're going to form a bond between two different ions, let's look here at how we form this ionic bond. So here we have a sodium atom and a chlorine atom. And the sodium atom has 1 electron in its valence shell, and the chlorine atom has 7 electrons in its valence shell. So chlorine is really looking for 1 more electron. And sodium, it's easier for the sodium to donate 1 electron to become stable than it is to accept 7 electrons to become stable. Because in order to become stable, it needs to have 8 electrons in that valence shell, right? So the sodium here has donated an electron to the chlorine atom. That has made the sodium slightly positive, so it's turned it into a cation. And then the chlorine becomes a chloride ion, and that's slightly negative. So now we have a negative chloride ion and a positive sodium ion. Because opposite charges attract, these are going to form what's called an ionic bond, which is a chemical bond that is going to keep them in close proximity to one another because they have those separate charges, and so they really like each other now, and they're going to hang out together because they've shared that electron. So I shouldn't have used the word shared there. The sodium donated the electron to the chloride ion or the chlorine ion. In covalent bonds, molecules actually do share their electrons, so that the electrons actually go around the valence shells of both atoms that are bonded together. So here on the top, we see hydrogen. So H2, we have 2 hydrogens. Each hydrogen atom has 1 electron in its valence shell, and so they each want 2 to make them stable. So they're going to share these 2 electrons. So these 2 electrons are going to now go around both of those protons and be shared. And that makes what's called a covalent bond. Covalent bonds are strong, and the more electrons that are shared, the stronger the bond is. So oxygen, oxygen has 6 molecules in its valence shell, so it needs to share 2 electrons with the other oxygen molecule. So that's going to be more stable or a stronger bond than the hydrogen bond where they're only sharing 2 electrons. So here you can see they're sharing we'll say 4 electrons, right? They're each donating 1 or sharing 1. Here they're each sharing 2. In carbon dioxide, we've got a carbon molecule and 2 oxygen molecules that are all sharing, they're each sharing 2 electrons here. So one more bit of vocab. A free radical is an ion or molecule containing unpaired electrons in the outermost energy level. So it's got an odd number of electrons in the outermost energy level. And free radicals are highly reactive. You've probably heard this term before. So free radicals in the body contribute to inflammation, they can contribute to cancer, all of those sort of negative outcomes. Last, we have hydrogen bonds. And hydrogen bonds are a weak bond that occur between hydrogen atoms and other negatively charged portions of a molecule. So hydrogen bonds cannot create molecules. They don't bring 2 atoms together. But when you have a molecule, they can change the shape of it. And we'll see this later when we're talking about DNA and RNA and proteins. We'll see how hydrogen bonds change the shape of proteins. So for right now, we're just going to introduce this, that hydrogen bonds are weak bonds that can change molecular shape, but they cannot create molecules. In water, hydrogen bonds are what cause the surface tension and sort of pull water molecules together. So if you see you have two droplets of water next to each other and they kind of move toward each other to create one big droplet, that's the hydrogen bond that's doing that. Chemical reactions in the body occur when new bonds form or old bonds are broken. And this is how molecules are formed or broken down. This involves reactants, which are substrates, so two things coming together to create a product, or when a product is broken down into its parts. Chemical reactions are really important in the body for growth, maintenance, repair, secretion. Secretion is like creating fluids, so we secrete hormones, things like that. Contraction, muscle contraction, providing energy to maintain homeostasis, like we talked about last week, creating proteins. Metabolism is the sum of all reactions taking place in the body at one time. So when we talk about your metabolism, we talk about all of the reactions taking place within your body, all of your cells and tissues simultaneously. We have two different forms of energy. Energy is the capacity to perform a work. Kinetic energy is the energy of motion, so to move an object to perform work, whereas potential energy is energy that is stored and has the potential to do work. And potential energy is what we store in our high energy bonds. Chemical energy is the form of potential energy that is stored in the bonds of compounds and molecules. So that's the specific name, is chemical energy, is energy that's formed in bonds. We have a couple different types of chemical reactions. We have anabolic reactions, which make things. When you think anabolic, I always think of anabolic steroids make big muscles. So anabolism is growth or building. Catabolism is breakdown or decomposition reactions. So it's the opposite of anabolism. And then we have exchange reactions where you are just moving or swapping one thing for another. You're not necessarily building anything or breaking anything down, but you're changing components. So here's our anabolic reactions. So in anabolic reactions, we are taking smaller molecules or atoms and assembling them into larger molecules. So here we are just, if you look at this, A plus B equals AB. So you're taking two separate substrates and combining them together into a product. So we're always forming new chemical bonds in anabolic reactions. Typically, we're using dehydration synthesis for this. And dehydration synthesis is where you create a molecule of water as a byproduct of the combination of those two molecules together. So here you can see that substrate A has a hydrogen attached to it, and substrate B has a hydroxyl group attached to it. So when we attach A to B, this hydrogen is going to bind to the hydroxyl group and create this water. And then the A and B are going to have a bond between them. So we're going to talk a lot about dehydration synthesis in different reactions throughout the day today. And so we want to always remember that dehydration synthesis is removing a molecule of water during the formation or attachment of two molecules in an anabolic reaction. So dehydration synthesis, always anabolic, always removing water. Catabolic reactions are breaking a molecule down into smaller fragments. So taking AB and separating it back out to A and B. And this occurs using hydrolysis. So hydrolysis is the opposite of dehydration synthesis. So with hydrolysis, you are adding a molecule of water to break apart the larger molecule into its smaller parts. So if we look down here, we have AB, and we're going to add a molecule of water. So the hydrogen is going to bond to the A, and the hydroxyl group is going to bond to the B, and that is going to break the bond between A and B and separate them back into their original parts. So that is catabolism, or the breakdown from AB to A plus B. In an exchange reaction, we are taking two different molecules and shuffling them around. So here we have molecule AB and molecule CD, and we want to detach A and B using hydrolysis and detach C and D using hydrolysis, and then connect A to D using dehydration synthesis, and connect B to C also using dehydration synthesis. So in order to do an exchange reaction, you're using both hydrolysis or decomposition and dehydration synthesis. Many reactions within the body are freely reversible, which means they can go in either direction, which is really helpful because sometimes we want to build things and sometimes we need to break things down to utilize their parts. So we show the reversible reaction using an arrow pointing in both directions, or it can be two arrows on top of one another, one pointing in one direction and the other pointing in the other direction, sort of half arrows. It's hard to create those in a PowerPoint presentation, but your book will show them. So at equilibrium, the amount of chemicals does not change, even though the reactions are still occurring. So things are always being broken down, things are always being built, but the overall amount of each chemical in the body is not changing when you're in an equilibrium state. Reversible reactions are always trying to seek equilibrium and balance opposing reaction rates. And so that's part of your body, is you always want to be in a homeostatic state, always trying to seek equilibrium. Enzymes are chemicals that can increase the ability of a reaction, can basically they're proteins that can perform synthesis and decomposition reactions within the body. So they promote these reactions by lowering the activation energy, so the amount of energy required for a chemical reaction to take place. Catalysts are compounds that accelerate chemical reactions without being changed or consumed. And most enzymes are considered catalysts. So an enzyme can be part of a reaction and then detach, and it remains unchanged. So here we've got our reactant and we've got our product. So without an enzyme, we need this much energy in order to create, to put these reactants into this product. But with an enzyme, we only need this much energy. So the enzyme is going to decrease the amount of energy required by the body to create this reaction. It also is going to make this happen a lot faster. So it allows our body to process things at the speed at which we need them. Each reaction has a specific energy involved in it. Some reactions create energy and some reactions utilize energy. Exergonic or exothermic reactions create more energy than they use. So these are common in the body and they produce body heat. Endergonic reactions or endothermic reactions utilize more energy than they produce. Nutrients are essential elements and molecules that are normally attained from the diet that are essential for proper functioning of the body. Metabolites are molecules that can be synthesized or broken down by chemical reactions inside our bodies. So nutrients we need to consume, metabolites we can create on our own. Organic compounds are compounds that always contain carbon and hydrogen, and the atoms are held together by covalent bonds. So in order to be considered an organic compound, you must contain carbon and hydrogen. Inorganic compounds usually lack carbon and hydrogen as primary structural ingredients, and they contain ionic bonds. And our body needs both organic and inorganic compounds in order to survive and function properly. Examples of organic compounds are carbohydrates, lipids, proteins, and nucleic acids like DNA and RNA. And we're going to go dive sort of deeply into all of those in biochemistry later on in this lecture. Inorganic compounds that are most important to the human body are carbon dioxide, oxygen, water, and then specific acids, bases, and salts like sodium chloride. We need both of those things. They're highly important in the body. Water is the most important constituent in the body, and it accounts for about 2/3 or 70% of your body weight. Any even small alteration of the amount of water in your body can be fatal, which is why severe dehydration can be fatal pretty quickly. As we already learned, water can be a solvent, meaning we can add water to molecules and break them apart through hydrolysis. So there are a lot of different organic and inorganic molecules that can be dissolved in water. If you think about adding salt to water and stirring it up, the salt will completely dissolve in the water. Water is also reactive, so body chemistry reactions occur in water. You have water throughout your body, inside your cells, outside your cells, everywhere. So hydrolysis is going to occur to break down molecules. Dehydration synthesis is going to bring molecules together. Water also has a high heat capacity, meaning that it can absorb a lot of heat without itself changing temperature greatly. So therefore, we can utilize water to carry heat throughout our body, dissipate heat out of our body without the water within our body really changing that much in temperature. So that's really important. A way that we stabilize our body temperature is we can carry heat to the surface and then allow it to disseminate or sort of radiantly heat out of our body and without our body temperature greatly changing. We also utilize water within the body for lubrication. We need to minimize friction between surfaces. We've got lots of different layers within the body of connective tissue, muscle tissue, our organs, our bones, and we don't want friction between any of these surfaces. So water is used as a lubricant in our joints, in our body cavities, between our organs, so everything can be close together, but not sort of getting stuck together. Water is also a polar molecule, so it's got a slightly positive pole and negative poles. And so inorganic compounds can undergo ionization or dissociation in water. Like we talked about, molecules can break apart into individual ions by hydrolysis in water. And then those ions can interact with the water poles, creating what are called hydration spheres. When you have an aqueous or water-based solution that has anions and cations in it, that can conduct an electrical current, especially when you have a membrane in between two aqueous solutions and you move cations or anions across that plasma membrane. This is how we allow our muscles to contract and our nerves to send impulses through our body to tell our body what to do. We're going to learn both of those in detail this semester, but it's nice to just put a little plug in for that now. Electrolytes are soluble inorganic molecules that can conduct electrical currents in solution. So sodium and chloride ions are some of our main electrolytes. And when your levels of electrolytes are disturbed, that can absolutely change all of your body's ability to function. So that's why we're really tightly regulated. They're regulated by the kidneys, by the digestive tract, and by the skeletal system. Calcium is another electrolyte that is highly regulated by the skeletal system and the digestive system. Hydrophilic compounds are compounds that like water, or we call them water-loving. So some of our organic molecules that have polar covalent bonds and ionic bonds are hydrophilic, which means they're attracted to water molecules. Hydration spheres can carry organic molecules into a solution, like glucose. So here's our water in our aqueous solution. So we've got our oxygen and our two hydrogens. So this oxygen is our negative pole, the hydrogens are our positive pole. And so our glucose can interact with these water molecules. And here's our sodium and chloride that have been broken apart in the water as well. Hydrophobic compounds are organic molecules that do not have polar covalent bonds. They are nonpolar, meaning they have no net charge, no positive or negative terminals. They are neutral. And polar or nonpolar molecules do not dissolve or form hydration spheres, so they stay like if you think about oils, if you were to put vegetable oil or olive oil into a cup of water, you would see that the oil would stay in its own balls or spheres and it would not dissolve into the water. That is because oils are hydrophobic. They are non-polar and they do not break down in water. So hydrophobic means water-fearing. So salts are hydrophilic, oils are hydrophobic. Colloids are a heavy solution that contains proteins or other large molecules, and they often appear opaque. So like milk is a colloid. Cream and thyroid hormone is also a thick sort of colloid material. The suspension is where particles in the suspension are larger than those in a colloid, so they don't actually sort of move throughout as much as the colloid does. So they're in the suspension, but over time they will settle out. Whole blood is that way, where blood cells will settle out of the plasma. Or if you were to put sand in water, the sand would go straight through the water and settle out to the bottom. When we're talking about blood, we have a test called the erythrocyte sedimentation rate, which is literally testing the speed at which blood cells settle out of the plasma. As we saw before, hydrogen atoms have one single electron. So their valence shell is their first shell with only one electron, and that makes them highly reactive. So in the body, they can break chemical bonds, they can change molecular shape, and they can disrupt cell and tissue functions. So hydrogen ions are not ideal in the body. We need them for some things, but we don't want to have an overabundance of them. pH is a direct measure of the concentration of hydrogen ions in a solution. So a neutral pH is where you have a balance of hydrogen ions and hydroxyl ions. So pure water is a pH of 7.0, and that is our neutral pH. The pH of human blood ranges as about 7.35 to 7.45. So it's just slightly basic. Acidic pH is less than 7. Basic pH is greater than 7. So pH of human blood is slightly basic. Here we go. So acidic, lower than 7, you have a high concentration of hydrogen ions and fewer hydroxyl ions. A basic or alkaline pH is above 7, where you have a low concentration of hydrogen ions and a high concentration of hydroxyl ions. So here, this is showing us the pH scale. So it goes from 0, which is hydrochloric acid or stomach acid, which is a ton of hydrogen ions, through vinegar is about a 2 or 3. Saliva is slightly acidic. Urine is anywhere from 5 to 8, depending. And then we've got blood, which is just above 7. Ocean water there's a lot of salt in that. It's pretty basic, about 8.5. Bleach is just under 10. Ammonia, 11 to 12. And then sodium hydroxide is 14. So oven cleaners, things like that are very, very basic. So like I said, the pH of blood is 7.35 to 7.45, and we try not to fluctuate outside of that. When you fluctuate far from that, you can start damaging cells and tissues. Chemical bonds are going to be broken. Protein structure is going to be disrupted. We have 2 different states that the body can go into. Acidosis, which is a blood pH below 7.35. Acidosis can happen with diabetes. Breathing difficulties can change your acidity as well, because carbon dioxide and hydrogen are linked to one another. So if you're not able to exhale your carbon dioxide, you can go into an acidotic state. Severe diarrhea, you can lose too much bicarbonate. And then alkalosis is where your blood pH is above 7.45, so you have too many hydroxyl groups. This leads to sustained muscle contractions that you can't control. And alkalosis can be, again, from respiratory disorders, lung disease, aspirin overdose, shock, anxiety, severe vomiting with loss of hydrochloric acid, diuretics, or overuse of antacids. Those can all send you into alkalosis. So this acid-base balance is very important within our bodies. An acid is any chemical that dissociates in solution and releases hydrogen ions. The release of hydrogen ions is going to lower the pH, and hydrogen ions are referred to as our proton donors. So proton, remember, is our one of the basic subatomic particles, and hydrogens can donate protons. Strong acids dissociate completely in solution, and the reaction moves in only one direction, so you cannot come back from it. So hydrochloric acid dissociates into hydrogen ions and chloride ions, and that is what breaks down food in our stomachs. A base is a solute that removes hydrogen ions from a solution and acts as a proton receptor. So in a solution, many bases release a hydroxide ion. The hydroxide ion is going to bind the hydrogen and form water. So they have a great affinity for one another. So anytime you have a hydroxide ion, it can bind a hydrogen ion and form water. Strong bases, like strong acids, are going to dissociate completely in solution. So like sodium hydroxide will become sodium and the hydroxyl group or hydroxide group, and the hydroxide can bind those hydrogen ions. Weak acids and bases do not completely dissociate. They're often reversible reactions, and they have less of an impact on the pH than the strong acids and bases do, but they do still impact it. A salt is an ionic compound consisting of any cation except hydrogen and any anion except the hydroxide group. They're held together by ionic bonds, and most of them completely dissociate in water and release their anions and cations. Table salt, which is sodium chloride, will dissociate completely in water. Buffers are substances that can stabilize the pH of a solution by either removing or replacing hydrogen ions. So they're typically a weak acid and its related salt, which functions as a weak base. So they can either accept hydrogen ions to increase the pH, or they can release hydrogen ions to decrease the pH and keep the pH where it needs to be. So buffers are very important within our body fluids to maintain our pH within a normal limit or normal levels. Organic compounds in the body always contain carbon and hydrogen, and they frequently contain oxygen. They have long chains of carbons linked by covalent bonds. Many of them are water soluble, with a few exceptions, and they're very diverse. But certain ones occur again and again, and these are called functional groups. Here's a list of our functional groups. So we have the hydroxyl group, the carboxyl group, the amino group, the methyl group, and the phosphate group. Hydroxyl group is polar, and we find these in all four types of organic compounds. They're involved in dehydration synthesis and hydrolysis reactions. The carboxyl group is found within fatty acids, amino acids, and many other acids. The amino group is found within amino acids, which are the building blocks of proteins. The methyl group is also found within amino acids. And the phosphate groups are found within phospholipids and nucleotides. Now we're going to talk about biochemistry. So biochemistry is the chemistry of our bodies. And this is how we're going to look at all of the organic components of our system. So carbohydrates are organic molecules that contain carbon, hydrogen, and oxygen in a ratio that is close to 1 carbon, 2 hydrogen, 1 oxygen. These include sugars and starches, and we have 3 different types: monosaccharides, which are simple sugars, disaccharides, which are 2 sugars connected, and polysaccharides, which are multiple sugars. Monosaccharide is a simple sugar. It's a carbohydrate that contains between 3 and 7 carbon atoms, and they're named for how many carbon atoms they have. So a triose has 3, a tetrose has 4, a pentose has 5 carbon atoms, and a hexose has 6 carbon atoms. So we're going to talk about hexose and pentose later. Glucose and fructose are both hexose molecules. And another vocab word, isomers, are compounds that have the same molecular formula but a different structure, and then they can be treated a little bit differently by the body. Here's our structure of glucose. So it can be shown in multiple different ways. So here we're just showing that glucose has 6 carbons. It's got 5 hydroxyl groups. It's got 7 hydrogen ions and 1 oxygen. Here it's shown as this hexose, it's a hexagon there. This extra carbon is off to the side with an oxygen in here. And then this is showing you with the actual molecules. Monosaccharides, like we said, are simple sugars with 3 to 7 carbons, like glucose, fructose, and galactose. Disaccharides are 2 simple sugars that are condensed together through dehydration synthesis, producing a molecule of water. Sucrose and maltose are examples of disaccharides. So before you can utilize a disaccharide for energy in the body, it has to be broken down through hydrolysis. So you must add water to it to break it down into its component monosaccharides, and then those can be utilized by the body to create energy. Polysaccharides are when you have many monosaccharides that are condensed together, again through dehydration synthesis. Polysaccharides include glycogen, which is how the body stores glucose for future use. This happens in the liver and in the muscles. Starches and cellulose are also considered polysaccharides. If you think about plants, if you're eating a starchy plant-based diet, there's a lot of breakdown that has to go into the cellulose of a plant before you can utilize that in your body. This is showing us the 3 different monosaccharides, fructose, glucose, and galactose, and how they can be combined in different ways to make 3 disaccharides. So fructose and a glucose together make sucrose, 2 glucose molecules makes maltose, and a glucose and a galactose makes lactose. Here we're joining glucose and fructose through dehydration synthesis. So you can see that the hydroxyl group from the glucose is going to join with the hydrogen from the fructose. And you'll end up with this bond at the oxygen here to create sucrose. And then you've created water as a byproduct. So to create that disaccharide through dehydration synthesis, you take out the hydrogen and the hydroxyl group to create water, and there in that process creates a bond between the 2 individual monosaccharides. Now, if we want to break sucrose down, we have to add a molecule of water through hydrolysis. And there we're going to have the hydroxyl group is going to connect into the glucose, and the hydrogen is going to bond to the fructose and separate those 2 molecules. Here we've got just a table that you can use for studying if you like, to show the different monosaccharides, disaccharides, and glycogen, what their functions are, and where you can find them. All right, we're moving on to lipids. So lipids are fats. They're mainly hydrophobic molecules like fats, oils, and waxes. They're made mostly of carbon and hydrogen atoms, and they include fatty acids, eicosanoids, glycerides, steroids, phospholipids, and glycolipids. Fatty acids are long chains of carbon and hydrogen with a carboxylic acid group at one end. They're relatively nonpolar, except the carboxylic group. Remember that nonpolar means that there's no net charge, and nonpolar compounds are hydrophobic. Fatty acids can be saturated with hydrogen, meaning there are no covalent bonds, or it can be unsaturated, where there's one or more double bonds. Monounsaturated means there's one double bond, and polyunsaturated means there's two or more double bonds. Anywhere you find a double bond, you're going to find a bend in the fat. So if we look at this picture, a saturated fatty acid is a nice straight flat chain, whereas an unsaturated fatty acid that has this double bond is going to have a bend in it. So saturated fatty acids pack together tightly, whereas unsaturated fatty acids cannot pack together so tightly. So they're slightly less bad for you. Eicosanoids are lipids that are derived from arachidonic acid, and they're absorbed from the diet. Two classes of eicosanoids that are important in the body are leukotrienes, which respond to injury and disease and are part of the immune response, and prostaglandins, which are hormone-like substances. Prostaglandins have a couple of different functions. They can regulate blood pressure, regulate inflammation, and sensitize nerves to pain. Non-steroidal anti-inflammatories, or NSAIDs, decrease the effect of prostaglandins, and so that's how they work to decrease pain. Glycerides. So lipids are composed of a glycerol, a 3-carbon backbone, attached to fatty acids. So glycerol has 3 carbons. Triglyceride has 3 fatty acid tails. Triglycerides are an important energy source. They're burned at night while you're sleeping. They're also can be burned during long, slow exercise. They function to transport fat-soluble vitamins like A, D, E, and K, provide cushioning for the body, insulate the body to retain heat, and they form components of cell membranes. So here we're looking at the formation of a triglyceride. So we have this glycerol, 3-carbon backbone, right? And then we have these 3 separate fatty acids. So these 2 are saturated with hydrogen. This one is unsaturated, so it has this double bond here. So we are going to take the hydrogen ions from the glycerol and then the hydroxide ions from the fatty acids. Through dehydration synthesis, we're going to create 3 molecules of water while attaching the 3 fatty acid tails to each of the carbons on the glycerol. So triglycerides are 3-carbon backbone with a fatty acid tail coming off of each of the carbon atoms. Omega-3 fatty acids are where the first double carbon bond is at the 3rd hydrocarbon from the methyl group. I can't go back to show you this, but it'll show you in your book. And so those are supposedly the best type of fatty acids for us. Okay, steroids are a foundation of 4 hydrocarbon rings bonded to a variety of different functional groups. Steroids, cholesterol is a main steroid. Cholesterol is a component of plasma cell membranes. It's very important in our body. We often think of cholesterol as being bad for us, but we actually do utilize a lot of cholesterol. We just also eat a lot of cholesterol, and so most people have excessive amounts of cholesterol in their body. Steroids are also important for sex hormones, estrogen and testosterone, corticosteroids, and calcitriol for regulating the metabolism, and bile salts are also derived from steroids. So here we can see cholesterol. So we have these 4 different rings, and then different functional side chains that make the cholesterol different from the estrogen, different from the testosterone. Diglycerides, remember 2 sugars bound together, 2 monoglycerides bound together, can attach to either a phosphate group, creating a phospholipid, or a sugar, creating a glycolipid. Phospholipids are extremely important in the body. They have a hydrophilic head and a hydrophobic tail, and these are what our plasma membranes are made out of. So the cell membrane for almost every cell in the body is made out of phospholipids. So super, super important. Glycolipids are also part of a cell membrane. They're just not like the integral part that make up the actual cell membrane itself. Here's a picture of a phospholipid. Looks like a person with very long legs, but you can see that it's got the non-lipid group. So this is going to be your hydrophilic head, and then the phosphate group, and then your fatty acid. So your glycerol with your 3 carbons, and then your 2 fatty acids. So this part is going to be your hydrophobic tail, and this is going to be your hydrophilic head. Glycolipids are going to still, again, be the glycerol with the fatty acids, but they're going to be bound to a carbohydrate. So this table you can use to study from, just showing you the different types of lipids, examples of each, what their functions are, and some information about them. Next, we're going to talk about proteins. So proteins are the most abundant and important organic molecules. And I cannot stress that enough, that the function of your cells is to create proteins. Our bodies are made up of proteins. Our bodies utilize proteins to do everything. Our DNA is a copy of the instructions to create every protein in our body. Proteins contain carbon, hydrogen, oxygen, and nitrogen. Notice that nitrogen is new and different. We have not seen nitrogen before as an essential element. And the building blocks of proteins are 20 different amino acids. Proteins are used for support, for movement. Our muscle is made up of contractile proteins. Transport, we have carrier proteins that transport things into and out of our cells. Buffers are made of proteins to regulate the pH of our bodies. Enzymes are made out of proteins. Hormones are made out of proteins. Antibodies are made out of proteins. Your body just cannot function without proteins. So proteins are long chains of amino acids, and amino acids all have a really similar structure with a variable side group. So they have a central carbon atom, a hydrogen atom, an amino group, a carboxylic acid group, and that variable side chain. So if we look in a picture, here's our central carbon atom, our hydrogen group, our amino group, our carboxylic acid group, and then this variable side chain that makes them different from one another. To create proteins, we are connecting amino acids together through dehydration synthesis. So one amino acid is going to connect its amino group to the carboxylic acid group of another amino acid. And that is going to produce what's called a peptide bond. So when you think of peptide bonds, think of proteins. We only find peptide bonds in proteins. So here we've got glycine and alanine, which are two separate amino acids. This is the carboxylic acid group of the glycine and the amine group of the alanine. We have the hydrogen and the hydroxyl group that are going to bind to one another to create water in dehydration synthesis, and we're going to form that peptide bond between the carbon and the nitrogen. Conversely, if we want to add water in, we can break this peptide bond through hydrolysis and separate the two amino acids from one another. Proteins come in multiple different shapes. So the primary structure is just the sequence of amino acids that create that polypeptide. So a polypeptide is a chain of amino acids prior to it becoming a protein. Secondary structure forms when hydrogen bonds form between different parts of the polypeptide, creating spirals or pleats. And we'll see a picture of this in a minute. Then we can have a tertiary structure where the secondary structure can fold more into a unique shape. And then the quaternary structure is the final shape of the protein where several tertiary structures come together. And the shape of the protein is extremely important. It is how the protein can function, what it does in the body is all connected to its shape. So here we have this sort of string of amino acids, and this is shown in that little area here. And then the strand is going to coil into the secondary structure, and then that coil is going to bend and fold into a tertiary structure, and then multiple tertiary structures will create the final protein. If we want to look here, this is our chain of amino acids, and then we end up with hydrogen bonds between, so see how amino acid 2 is going to bond to amino acid 6. So when that pulls them together, 3 to 7 and 1 to 5, this is how it's going to end up spiraling them. So remember, hydrogen bonds cannot create molecules, but they can change the shape of compounds. So they're changing the shape of this protein, but they can't create different molecules. Here's our tertiary structure of a heme unit. And when we put multiple heme units together, we form hemoglobin, which is a globular protein. And that's our quaternary structure. Fibrous proteins come in sheets or strands, like collagen. We'll see that in a couple of weeks. Collagen is the building block of muscles, not muscles, of basically all of our connective tissues are made up of collagen. And so it's collagen are strands, but they can come together to form sheets. And then globular proteins are, like they sound, spherical. And they are more active. So they'll have like enzymes, might be a globular protein, things like that. And so their shape is based on the sequence of amino acids and the hydrogen bonds that make them fold into new shapes. So here's the difference between a globular protein and a strand-like protein. Enzymes we talked about earlier, so we'll just touch on them again. Enzymes are proteins, that's why we're going to talk about them again. So they are catalysts, meaning that they are there to speed up reactions by lowering the activation energy of a chemical reaction. In that process, they are not changed or used up. They simply are there to support a chemical reaction and then detach and go on to do it again. They're very specific, meaning they only work with a limited number of substrates. They can become saturated, meaning you can only have so many things bind to them at once. And they are regulated by other chemicals within the cell. So here in pictures, we've got two different substrates that we want to bind together. So here's our enzyme, and you can see that there are two binding sites on the enzyme. This is the specificity. So this is a rounded attachment, and this is a pointed attachment. So you can see the rounded active site here for substrate 1, the pointed active site here for substrate 2. So the two substrates come in attached to the enzyme. This puts them in the perfect spot for them to interact with one another, creating the final product. And then the final product releases, and the enzyme is unchanged and now able to go do this whole thing again with new substrates. But you can see if there's only two active sites, it is now completely utilized. There are no other active sites allowing it to produce more product until that product releases, and it can then undergo this situation again. Cofactors are an ion or molecule that binds to an enzyme before a substrate can bind to it. So sometimes an enzyme needs something else to bind to it in order to make it capable of binding to the substrates. So that's a cofactor. A coenzyme is a cofactor that is a non-protein cofactor. So like a vitamin can be a coenzyme. Denaturation occurs with heat or with changes in pH. So proteins can be broken down with heat, meaning they lose their shape. And when they lose their shape, they lose their function, because remember, their function is highly tied to their shape. So loss of shape or function with high heat or with changes in pH. So an acidic environment or an basic environment can change the function of proteins, and enzymes are proteins. This heating up an egg is a great example of that. So eggs are basically all protein. So if you think about an egg being sort of fluidy when it's raw, and then you put it in a hot frying pan and it gets firm, that is what happens. That is denaturation of the protein. We can also have combinations of proteins and carbohydrate molecules. So we can make glycoproteins, which are a large protein with a small carbohydrate. And these include enzymes, antibodies, hormones, and mucus production. People often think of mucus as being just something disgusting that happens to us, but actually mucus is super important in our body as it coats our reproductive system, our digestive system, and our respiratory system. So it gives a level of protection and lubrication of all of these systems. And then we have proteoglycans, which are a large polysaccharide, so a large sugar attached to a polypeptide or a chain of amino acids. And proteoglycans promote viscosity. Viscosity is when a fluid is syrupy, almost like a molasses, or if you think about a raw egg, that is sort of a viscous, what's the word I'm looking for? A viscous feeling to it. So proteoglycans are going to help with coating, keeping our fluids from, man, I'm really struggling with this one. They help with keeping the fluids of the body a little bit thicker and slippery so that our surfaces don't stick together. All right, our last organic molecule are nucleic acids. So our nucleic acids are large organic molecules, and they are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. So they store and process molecular information inside of our cells. And our two classes of nucleic acids are DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid. These classes differ in composition, structure, and function. So DNA is what determines our inherited characteristics, our eye color, hair color, our blood type, our body shape, the way that we grow. If we have a mutation in our DNA, we're going to have a disease. All of those things are controlled by the DNA. RNA is assisting with protein synthesis. So it basically makes a copy of the DNA instructions on how to create a protein or all of the proteins, and then takes that copy out of the nucleus of the cell into the cytoplasm where these proteins can be assembled. Nucleotides are the building blocks of DNA and RNA, and they have three parts: a sugar, a phosphate group, and a nitrogenous base. The sugar is a 5-carbon sugar, which is a pentose, and that's either deoxyribose or ribose. So that's the difference between DNA and RNA, is the 5-carbon sugar that is connected, or that makes them up. They both have a phosphate group, and then the nitrogenous bases there are 5 different nitrogenous bases. So adenine, guanine, thymine, cytosine, and uracil are the 5 different bases. Thymine is only in DNA, and uracil is only in RNA. So here we're looking at a nucleic acid. So we have our pentose sugar, our phosphate group, and then our nitrogenous base. So this might be adenine, it might be uracil, but that's our nitrogenous base. Purines are adenine and guanine, and purines have 2 ring structures here, which accommodate several nitrogen atoms. So adenine and guanine are our purines. Pyrimidines are cytosine, thymine, and uracil. And like I said, thymine is only in DNA, uracil is only in RNA, and the pyrimidines only have a single ring structure. That's not going to be the biggest of deals. You don't have to necessarily memorize that, but do know which ones are purines, which ones are pyrimidines, and which one is only DNA and which one is only RNA. DNA is double-stranded, and the bases form hydrogen bonds to hold the DNA together. RNA is single-stranded. DNA forms a double helix, and complementary base pairs are going to hydrogen bond to one another. So purines are going to pair with pyrimidines, and each purine has a specific pyrimidine that it pairs with. So in DNA, adenine always pairs with thymine, and cytosine always pairs with guanine. In RNA, uracil replaces the thymine because thymine cannot fit out of the nucleus through the nuclear pore. So the uracil has to be substituted into the RNA so the RNA can leave the nucleus of the cell and move out into the cytoplasm. So in RNA, we would have uracil. So to make a little bit more sense of that, when we have our double helix, we have hydrogen bonds between adenine and thymine, right? So they are bonded together, and our phosphate group and our deoxyribose are on the outside here. So this is our double helix where we have 2 strands that are hydrogen bonded together. So adenine and thymine are always going to be our complementary bases, and then guanine and cytosine are our complementary bases. Okay, so cytosine and guanine. So that's how our DNA works. And then our RNA molecule is a single strand, and so it's got uracil in it instead of thymine. And we'll see next week more information about how proteins are synthesized and how RNA copies DNA and all of that fun stuff. So our types of RNA, we have 3 different types. We have messenger RNA, transfer RNA, and ribosomal RNA. And these are all different shapes. So the one that we just saw, that single strand, that's messenger RNA. And next week, we're going to look again more closely at transfer RNA and ribosomal RNA. So here are our nucleic acids. So you can use this just as a comparison between RNA and DNA for study purposes. All right, so cellular energy is, we need energy to perform our vital functions. And our cells are going to obtain this energy by breaking down organic substrates or catabolic reactions. And then this energy must be transferred for use by cells. Different parts of cells require energy to power specific actions and reactions. So the method of energy transfer involves the creation of high energy bonds by enzymes within cells. And then when those bonds are broken, that energy is released and can be utilized directly by the cell. Nucleotides that can be used to store energy are ADP, adenosine diphosphate, and ATP, adenosine triphosphate. So ATP is what we typically think of when we think about powering the cell. Phosphorylation is the adding of a phosphate group to an ADP to create an ATP. So it's just a vocab. I want you to know what phosphorylation is. So phosphorylation is basically adding a phosphate group. So let's look at this in pictures. Here we've got an adenine and a ribose. So this is adenosine, is an adenine connected to a ribose. When we add one phosphate, it's called adenosine monophosphate, or AMP. When we add a second phosphate, that is the high energy bond, and that makes adenosine diphosphate, ADP. When we add a third phosphate, we have another high energy bond, and that is ATP, adenosine triphosphate. So we're storing a lot of potential energy in the bonds between the phosphate groups of the ADP and the ATP. So if we want to break these down, we can remove a phosphate and release the energy that's stored in this high energy bond and utilize the energy in the cell. So all of these different building blocks that we've been talking about form cells or functional units within our body. And then our cells differentiate to do different things. So we have muscle cells, we have nerve cells, we have heart cells, we have all these different kinds of cells. But metabolic turnover allows our bodies to grow, to change, to adapt, and our body recycles and renews all of its chemical components at different intervals. So this shows you what the inorganic and organic components of the body are. So like water is our main inorganic component. What its building blocks are, hydrogen and oxygen. What its sources are, absorbed from the diet or generated by metabolism, right? Dehydration synthesis generates water. And then what are its main functions? So this is a nice study tool for you to look at. What are all the components of the body? What are they made out of? Where do they come from? And what do they do? And then this is just a little, you don't have to memorize this, but this is just interesting to show that in the liver, we have Protein that is recycled every 5 to 6 days. Enzymes can be recycled every hour or several days, depending on the different enzymes. Glycogen, which is stored glucose, is recycled every 1 to 2 days. Cholesterol every 5 to 7 days. Muscle cells are renewed every month. Glycogen in the muscle every 12 to 24 hours. In neurons, our phospholipids are renewed every 200 days and cholesterol every 100 days. And in fat cells, triglycerides are replaced or recycled every 15 to 20 days. So that's the end of our lecture. Hopefully you've learned a lot this week. I know it's a lot of new information, but I promise you we're going to build on all of this information throughout the rest of this semester and next semester. So getting a good foundational understanding now is totally worth it. Have a great week.